Hello everyone, and welcome to the first official video of S&E, where today we are going to be talking about the structure of muscle. So to get things started, I'm just going to quickly differentiate the difference between muscle structure and muscle architecture, because there will be a different video on muscle architecture probably next. But yeah, I just want to quickly differentiate the two. So muscle structure, I'm mainly talking about the different layers of muscle. So you'll see in a second about this image up here. I'll go through that. And then we're also going to talk about sarcomere, what's inside a sarcomere, how the sarcomere contracts, how muscle contracts. So that's what I mean by muscle structure. And then muscle architecture is more going to be about different ways muscle can be built up. So in that episode, we're probably going to be talking about muscles that have more sarcomeres in series versus parallel, or vice versa. And then maybe like different types of muscle architecture. So like fusiform, unipennate, etc. So we'll get to that in a different episode. Just wanted to clearly differentiate the two before we move forward. So we're good? Perfect. So if I could direct your attention to this image on screen here, hopefully you can see it clearly. So I'm going to walk you through this one to start with. So muscle itself as a whole is made up of B. Yes, the correct answer is B, fascicles, right? So muscle itself, the belly, you can see here, these are fascicles, right? And what epimysium does, the very outer layer, it has a role in making sure these fascicles are all bundled together nicely in what we call a muscle, right? So now, delving a bit deeper into this, these fascicles, so as you can see here, this obviously is a fascicle, and this here is like it pulled out. So we're going deeper as this comes pulled out. So this fascicle, they're made up of a bunch of fibers. So this is a fascicle here, and you can see in this a bunch of little fibers. And that is the fibers themselves, the fascicles are surrounded by the perimysium. So these individual fascicles are surrounded by perimysium, and then all those fascicles are bunched together and surrounded by epimysium. All right, I hope you're still with me. So now, as we delve a bit deeper, you can see yet again we've kind of pulled this fiber out, so we're zooming in even deeper. So this is an individual fiber, and these fibers are made up of many myofibril, and you can see that here. You got lots of these little tiny myofibrils, and those myofibrils, I guess, are the smallest. And then inside these myofibrils is the contractile unit itself, the sarcomere. So these little tiny things have many sarcomeres in them, and they consist of the contractile element of muscle. So that's it for now. So what I'm going to do quickly is just write up on the board my way of remembering. So I guess if you would like, this could be the first official tip.
Woo! Yeah, baby! That's what I've been waiting for.
Of the S&E channel. And this is a little trick that I use to remember between epi, peri, and endomysium. So for starters, they've all got mysium in the name. So to some degree, that makes it a bit easier for us to remember, because then all we have to worry about now is remembering just the first three letters. Or four in the case of the peri. So how I like to remember is I remember that epi is the very outer layer, and endo is the very deepest layer. And then how I remember that, right, is end kind of to some degree means the same or is related to deep. So I remember that endomysium is the deepest layer. And then I remember as well that the other E, epimysium, is the outer layer. So then all you have to do is just remember that perimysium is in between those two. Perfect. So that's what we've got so far. Just quickly before we move on, I'm just going to quickly draw a very rough myofibril, just so we can get that covered now before I move on, because I want to make sure that hopefully there's no confusion within the first 5 minutes. That would not be good teaching on my behalf. So what we're going to do is just draw this. So that essentially is what a myofibril is, right? Just like this line. It's a rectangular line, box, whatever you want to say. So this is one very, 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 very small portion of a myofibril. And then within this very small portion of a myofibril, we might have boom, 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 boom. We might have 4 or 5 sarcomeres. So these are sarcomeres. So they just cover up a very small portion. They're adjacent to one another in a myofibril. So that's essentially what we're talking about when we mean myofibril. And these sarcomeres themselves are the contractile elements. So now talking about the sarcomere, right? Oh, geez. I apologize, guys. This whiteboard has seen better days. Maybe a future investment. Anyway, sarcomeres, right? So we're talking about 4 major proteins that lie within the sarcomere that are responsible for contraction. So I'm mainly just going to focus on the most important 2, which is that being actin and myosin. The other 2, we've got tropomyosin and tropopin. I don't know if I've pronounced that last one right. Not the best with pronunciation. So those last 2 I mentioned are basically just side characters to actin and myosin. So they're just going to basically either help activate or help prevent the interaction between actin and myosin. So they're not as major, I guess, as actin and myosin. And for the sake of simplicity and the fact that I am still a uni student, I'm just going to go over actin and myosin because they play a much easier role for me to explain off the top of my head, right? So actin is also referred to as a thin filament, and myosin being the thick filament. Okay, guys, we have got the sarcomere. I know, isn't it kind of messy? Not really that neat. That's my bad. I'm not the best drawer, and that is exactly why I've got this handy little printout here, because I'll tell you something for free, guys. I'm not drawing that. I can't draw that. So, yeah, my apologies. Anyway, so we've got the sarcomere here. Quick little fun fact first, right? So we've got here this little sentence, if you'd like. 3,700 sarcomere per centimeter of myofibril. So I was just mentioning before how a very small portion of a myofibril might have this many sarcomere. Well, there you go. That's the exact number. So what I drew before must have been a very, 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 very microscopic piece of myofibril to only consist of 4 sarcomere. Anyway, I'm getting off track. I'm yapping. So I'm going to explain this because you've probably been looking at it a bit weirdly for a while now. So I'm going to start with these blue little zigzags. Believe it or not, they are called the Z line. So the sarcomere consists of 2 Z lines, and that can kind of be how you mark them, I guess. So from one Z line to another is one sarcomere. So then we've also got sarcomere on this side and this side. I've just drawn them in very roughly because I'm not going to talk about those ones. This is the important one here. So these green lines that come out and stop before the middle is actin. You might remember that I mentioned not too long ago that actin is the thin filament. And this middle part here, well, I'm hoping you can guess what that is. Yes, if you said myosin, that is correct. Myosin being the thick filament. So this myosin kind of takes up this middle part where the actin stops, and I guess slightly overlaps where the actin is. So now that we've got that down pat, hopefully there's no confusion. Well, I'm going to talk about how these things can make a muscle shorten and how you can do a bicep curl, for example. Like what? How does that happen from this? Well, it's a very good question. Let me explain it to you. So, essentially what's going to happen is these filaments are just going to slide together. Pretty simple. So these Z lines are actually going to move slightly closer to one another, while these filaments actually do not change in length. Very important. They do not change in length. That is why this is called the sliding filament theory. Okay? So they're just going to slide past each other. There's going to be no change in length. Awesome. So what's going to happen is this bad boy here, the myosin is going to basically fish over to the actin, and it's going to grab on to an active site on actin. They have many of these active sites. It's going to grab to an active site and just kind of wind it in, pull it closer to the middle, and they slide past each other. And that is contraction. So this actin might get pulled to here, and this one might get pulled to there. Boom. Easy. They've shortened. Great. That's how it works. That's the sliding filament theory, essentially. They slide past one another. So this model is still, to date, pretty validly correct. But the problem with this model, right, is that it cannot explain all actions of a muscle. A really great example of this is you could probably try this yourself, but you may notice that you are very weak when your muscle is very short. So when you're doing a bicep curl and you've come to around 30, 20, I'm not quite sure. I don't have a, what's the one, protractor? Something like that. I don't have that to measure the angle here, but this is, my bicep is flexed, right? So the sarcomere, the muscle itself, is very short. So we're really weak in this position, right? And that is because these bad boys are moving from their optimal length. I'll explain that in a bit, don't worry. But what happens is when you lengthen, right, you would think you would lose a lot of force similarly to when you go too short. That is correct. However, when you lengthen a muscle, you actually retain more of that force than when you contract the muscle. So this model itself can't actually explain why you're stronger lengthening a muscle than you are when you are really short. So that is where one of my good personal friends in my body, personal friend, Titan, comes into play. So I'm just going to draw it here, blue dotted line. So I guess it kind of plays in with the Z line, but this is not the Z line extending or anything. This is Titan, the largest protein in muscle. And what Titan is going to do is it basically anchors myosin to the Z line. So there you go. It's kind of related to the Z line. So they're on both sides, by the way, and on all of them. I just try not to waste too much of your precious time. So what's going to happen here is Titan is going to resist lengthening. Boom. Pretty much right there. I hardly need to say anything more. So, but I will, don't worry. So as the muscle lengthens, Titan is going to kind of stretch out and it's going to resist this stretch as best as it can. And that itself is going to generate some force, some energy for you to use. And that simply explains how you stay strong when you're lengthening. But, always but. There is more to this than just this. There are other components of muscle that help tighten out here. And I am going to go over them in this video as well. But there are also other ones that I'm not going to go over in this video. Okay? Alrighty guys, we are going to get another printing on the board. Yes, I know, I've intentionally put it the wrong way up because I want it to be a secret as I chuck it up. Ready? Boom. Okay. So, this could look a bit scary to you. It looks a bit scary to me, don't worry. I'm still only halfway through my second year. But I'm not going to go into too much depth about this. I just want to highlight one very crucial point with this image here. And that is actin, myosin, right? Look at actin. Actin looks Like what? What are these balls on there and like little vines going through them? What? This is the point I want to showcase to you. Actin is a very, very complex protein. Myosin, not so much. That's it. That's all you need to pick up from this image. That's all I really understand from this image. That actin is very complex, myosin is not as complex. So that's great. And that is purely just because actin is the binding site. So it has to be more complex. It's got all these binding sites. And tropopin and tropomyosin, which I said I'm not going to mention, but I might just quickly here. Basically, it's going to help prevent binding to actin when it's not necessary. So yeah, and that's why there's kind of these vine thingies there. They help like lock up the actin. But I'm not going into that, okay? That's all I want to highlight here. That actin is very complex, myosin not so much. But I do want to talk a bit more about myosin because myosin is fascinating, right? So you might notice on this image here that myosin's got like this little head and like these little tails that kind of tangle up, I guess. So if I just draw that here, it would simply just look like, you know, got this green head and this red tail. Don't know if they're actually green and red, but who knows. Anyway, quickly, this green part is going to be called the S1 segment. That is what I'm going to refer to as the myosin head for the rest of this video, okay? S1 equals myosin head, S1 segment. And then this red tail is going to be the S2 segment, not the SZ, sorry, the S2. The S2 segment. And once again, that is what I'm going to refer to this as for the rest of the video. So no myosin tails. Okay, cool. So what is going to happen here, what I'm going to quickly show you, is that by themselves, myosin is just like this weird kind of thing with a head and a tail, with an S1 and an S2. But what happens is if you get lots of these myosins together, as you can see here, there's just two of them, but they like wrap around each other. And I'm going to put an image on screen, don't worry, of what a full myosin filament would look like, because these are just myosin molecules. So they wrap around each other and you get like this big kind of like, I guess, rope, weird looking rope. I'm just thinking if rope's the right word. Yeah, kind of like a rope, but it has properties other than a rope, I guess. More properties of like a rubber band, like a really strong rubber band. So I guess you could even say like just many like rubber bands wrapped together. And I'm going to get to that because this S2 segment has some elasticity involved, and that's where the rubber band analogy comes in. So I'm going to do a drawing up in a second, but I just want to quickly say that similar to the Titan, this S2 plays a good role in the lengthening of a muscle and being able to generate some force as the muscle is lengthening, right? So, yeah, I'll get to that drawing. Okay, nice big drawing that I'm hoping you can see. So, what I'm trying to showcase here is this elasticity within the myosin, the S2 segment specifically. So here we've got a very overlapped kind of cross bridge, right? So with this, you're going to imagine that, I'm just going to quickly draw here, right? So go back to like the Z line thingy. I'm not going to fully draw the Z lines up, but basically here, they're too close, right? So these actin is like very close, like to the point where they're almost like overlapping. Obviously it's impossible for them to actually overlap, but remember earlier when I was doing the Z lines? So this is what the sarcomere would look like in this case here, right? So this actin is very close to one another, right? So when they're close, there's not that many cross bridges that can be happening at a single instant, and the cross bridge connection is pretty weak, as you can see here, a very weak connection. And then also this S2 segment is very short, and that's because this itself is very short, right? So it can't be long. It's got to be like squished up, right? It's like all crammed. That's what this is, right? So then we get to this second one here. You know what? I like the idea of this, so I'm going to do it again. Very rough drawing, guys. I do apologize. But basically, in this next image here, This is showcasing like optimum cross bridge, optimum interaction between actin and myosin. Okay, so that optimum might be like there. There is like actual diagrams and stuff to show optimum lengths, but I'm going to get to all that. All this optimum force stuff is going to be in another video. Okay, I'm just quickly showing you here because I want to highlight this S2 contribution. So this is what optimum length might look like, right? So they're not too close, but they're not too far apart. So you can have this perfect connection here onto actin, and that is where you're going to get most force. Fun fact, because why not chuck it in? For the bicep, of course, that's going to be around about a 90-degree angle. So there you go. So that's that bad boy there. All right, and sorry, as you can see, this one, oh, let's see actually. They're about similar length, but this one's obviously going to be a little bit longer because it's not as short as this one. So just, you know what, I'll do it. I'll just draw this a little bit longer. So this is optimum length. And there's going to be a little bit of contribution, I guess, from the S2 because it's a little stretched out, but not as much as this one here, which I'm going to go into right now. So this is going to look like, you know what, I'm going to draw it further apart as well just to showcase, because the Z lines do move apart as well. So it would be more scientifically accurate if I did it this way. So what this might look like is now, oh no, that's not good. Now the actin is very, very far away. It's going to be hard to have a good connection and generate some force when it's that far away. You can see that here as well. Bad boy is pretty far away. But the good thing about this is, look, oh, now you've got this really long S2 segment, so it's stretching. It's going to resist that stretch, but at the same time, store energy in here. So that way, when it shortens again, you get this massive amount of energy that can be used in here as well. Not massive, but an amount of energy. I'm not really going to go into stretch, shorten, cycle stuff now, but that does happen as well. But the main thing I want to say here is that this lengthening is giving us force. It's resisting, but it's also storing energy in this state. Okay, you don't have to remember the storing part, but you just got to remember that this is helping us here in the lengthening phase. Similar to Titan, but I'm assuming Titan would do a better job than S2. Okay guys, to finish off, I'm just going to show you some quick and simple muscle models to kind of go into a bit more of potentially a practical implication of what I was just teaching. But before I delve into that, I just want to show an even simpler model here with these weird looking drawings that I've drawn. Yes. So I just want to clarify this very simply before I go deeper. So this is going to represent a muscle for the time being, and this is going to represent the elastic components. So I haven't mentioned this yet, but tendon also has elastic properties. The myosin S2 that we were talking about. So yeah. So what I'm trying to showcase here is as the muscle shortens, so does this elastic component, okay? And similarly, if I drew it up before, if a muscle lengthens, so does its other components, okay? Perfect, we've got that now. I'm gonna delve into a bit deeper. Okay guys, welcome back. Some confusing and also sloping down drawings. Wow, I didn't realize that until just then. My apologies. So I'll quickly explain this. So up here, we've got this writing that was still here in the last frame as well, which I kind of teased you with, I guess, because I didn't explain it then. So that's going to represent this model that I'm drawing now. So CE in red is the contractile element. So that's going to be actin and myosin, specifically the myosin S1 segment, okay? S1 segment and actin, just that cross-bridge interaction. I don't know if I mentioned that earlier, actually, but a cross-bridge is simply just actin and myosin interacting. That's a cross-bridge. SEC, the series elastic component, that's in green here. that is going to be your tendons and your myosin S2 segment, okay? Because remember, both of those have elastic abilities. And those are in series because they're in series with the muscle themselves, okay? And then PEC is the parallel elastic component. And that is specifically just the mysium, epi, endo, peri, mysium, okay? And that's parallel to the muscle, right? So that's where the parallel elastic component comes in. And they have elastic capabilities as well. So, these little Zs in there represent that it's like shortened, right? Because if the Zs weren't in there, it would be just as far as it could go, right? If that makes sense. So as you can see here, this is fully lengthened, and that's got some Z in it, so it's got some slackness. So it's shorter, it's trying to like compose itself. So if you'd like, now that I've explained that, you can pause the video for a second and try and guess, based on what I've taught, how these different scenarios are going to play out. Alright, for those of you who stayed, or I guess paused, doesn't really matter, you both would be watching this, I'm going to explain a few scenarios here. So initially just relaxed muscle. So we've got a contractile element that is just relaxed, series elastic components that's relaxed, parallel elastic component that's relaxed. And we can kind of refer back to here if we want to see the differences. So isometric, right? What's going to happen is our contractile element is going to shorten, but the muscle as a whole won't. How's that happen? Well, series elastic components are going to lengthen, and that's going to kind of cancel out the movement. So that's very simple. And of course, in the parallel elastic component, there's a few Zs missing, so that's also going to do a bit of lengthening, but not too much. Now, for this passive stretch, right? So this is just lowering your bicep without any weight, not trying to resist it at all, just simply lowering it. So what's going to happen is your contractile element is going to be switched off. It's not going to be active. Your series elastic component, see there's a few Zs in there still, but not as many as up here. So that's because since there's no cross bridges, because the contractile element is switched off, it's not trying to resist it. So there's no interaction between actin and myosin. That's a cross bridge. So there's no cross bridges, which means there's no S2 contribution. And S2 is one of the series elastic components. So this slackness that has been taken up, this lengthening, is purely just from the tendons, okay? And we're going to get a lot of lengthening from the parallel elastic components because the muscle is still lengthening and somehow that's got to happen, but this bad boy is not doing anything. So parallel elastic component and the series is going to contribute slightly. But remember, not the S2 segment because it is not active. And then with an active stretch, so this might be holding a dumbbell and like trying to control the lowering, or even if you simply are just trying to resist the lowering, right? So of course the contractile element is going to be active this time. So that means this SEC component is going to be fully stretched out because now we've got the myosin S2 segments contributing and the tendons. Now the PEC, I've still drawn a little bit of a Z in it, and that's because when there's active lowering, it's not going to contribute a great deal. Like it is going to contribute a little bit, but not a great deal. So it is still going to lengthen, but not to a significant amount. So that's where that little bit of slackness, I guess, is still there. But active, this whole entire contractile element and the series elastic components, they're going to get their work done out for. They're going to be tested out here, okay? That's what's going to happen there. Okay, I'm going to finish up now because I've been going for a while, but I'm just going to quickly highlight one more point that I think is important and I missed it earlier on. So you might recognize this bad boy from earlier on. If you said that's a Myofibril, you would be correct. 5 gold stars for you. Now, what I want to say is obviously these little areas here are sarcomeres. So what happens, right, is as a sarcomere contracts as they slide past another, the force from that is going to be spread to adjacent myofibrils. So adjacent means left, right, and up and down. So for the sake of validity, I should draw another one because this is what happens in a muscle. You get myofibrils next to each other. as well. And what's going to happen is, say this bad boy is active, it's going to spread its forces this way, up and down as well. So we can get a lot of force generation happening simultaneously if all these bad boys are just instantly shooting action potentials everywhere. So that's how we can get like simultaneous contraction of a muscle. And that is also important for, there's experiments done when some muscle was sliced 50% in half. However, only 20% of strength was lost. So the explanation for that would be that the force is transmitted in a much greater way than you could ever cut muscle. So that is really fascinating. I'm not going to delve into that too much. It's not really important. But there you go. Thank you for watching my first video. If you made it this far, good on you. I apologize if the drawings were a bit messy and whatnot. I'm learning, so hopefully as I keep smashing out videos, the content will get better and better. But I would think that my drawings are never going to get better, so you're probably stuck with that. But lucky, there's such thing as printers, right? And they're going to be very helpful in this channel. Thank you guys so much for watching. Stay tuned, like and subscribe, and I hope you learned something today. Thank you.